Right, so we have made the concept quite clear over here. So now let us look into the first molecule, that is the amino acids. Now, amino acids are the building block of the proteins. Okay, yes. So if you look into any type of protein, ultimately they are made up of the amino acids, right? Yes. Now, why they are called as amino acids? Because they do have a very interesting property, that is the amino group, which is actually basic in nature, but whereas the same molecules also has another group, that is the acidic group. Okay, acidic group, and this acidic group is generally the carboxyl group in the case of the amino acids, right? So also you find simple R groups. You also find R groups, right? So we will discuss about the amino acid in the structural point of view. Okay, so if you have to draw a simple structure of the amino acid, it is NH2 C, that is carbon, and H, and here we are having the R group, and finally we are having is the carboxyl group, right? So this is the what we can say general formula, right? This is the general formula for the amino acids. Now here you can see that this carbon, what we are mentioning over here, is called as the alpha carbon. Okay, the reason why it is actually called as the alpha carbon, because you can see there are 4 different groups attached to this single carbon, right? So hence, as mentioned over here, it is an as amino group, which is basic in nature, and also the acidic group, and it has an alpha carbon, and for this alpha carbon, the 4 different valence groups are actually attached, right? For example, the first group you can see over here is the simple hydrogen, and the next one we are having is an amine group, that is the NH2 group is called as the amine group, and this group is mainly giving the basic property to the amino acids. And also the last one we are having is the carboxyl group, that is COOH is actually the carboxyl group, and this carboxyl group is giving the acidic property to the amino acid, as you can look over here, right? And here we are having one another group called as the R group, right? See, why do we use the alphabet R? R can be any group. Okay, it is just an alphabet that we're using to indicate presence of any group over here, presence of any group over here. Remember this, right? See, in some cases you find a simple hydrogen. Okay, let me draw or write one amino acid over here. Okay, NH2 alpha carbon, hydrogen, and carboxyl group, right? And let us substitute this R value as a hydrogen, right? So if you find a simple hydrogen, a simple single molecule of hydrogen in the R group, then this amino acid is the glycine, right? Now glycine is considered to be one of the simplest amino acids among all other amino acids, right? The first simplest amino acid that you can learn to study, right? We can even glycine, in the case of glycine, it is having simple hydrogen in the case of R group, right? So now in the case of another amino acid, if you consider alanine, okay, in the case of alanine, the R group is simply methyl group, right? That is CH3 group is actually a methyl group, right? And in the case of serine, okay, in the case of serine, you can see it is the R group is CH2OH, right? CH2OH, which is an hydroxy methyl group. So with the same skeleton of the basic formula of amino acid, you can just go on replacing the R group with the desired substituent group. You will get a different type of amino acids, right? So generally you see that in nature the amino acids are around 20 in number. Okay, so whatever proteins you consider, okay, so ultimately they are made up of the repetitive units of these 20 amino acids only, right? And the R group basically varies. So let us look into some other chemical properties of the amino acids, right? See, for example, we are having some amino acids which are charged, okay, which are charged. For example, there may be a positively charged or they may be what? Negatively charged amino acids. An example for negatively charged amino acid is glutamic acid. It is glutamic acid, right? And negatively charged amino acids are generally called as the acidic amino acid. But the positively charged amino acid, we are having an example called as the lysine, right? Lysine is an example for positively charged amino acid, and positively charged amino acids are actually called as the basic amino acids. And in some cases, you also find neutral amino acids. You also find neutral amino acids, right? An example for a neutral amino acid we are having is the valine. Okay, valine is an example for the neutral amino acid. And in some cases, you also find aromatic, okay, aromatic amino acids. We are having three examples that has been mentioned in your textbook. The first is the tyrosine, okay, tyrosine tryptophan, okay, and we are having one more, that is the phenylalanine, right? It is a very simple amino acid, alanine with a phenolic group. It gives us the phenylalanine. The name itself indicates, just attach a phenolic group to the R group, then you get phenylalanine. So this is some basic examples that we are having regarding the amino acid types based on the chemical properties of the amino acids, right? Now, there is also an interesting property, that is the amino groups and the carboxyl groups are actually ionizable, okay? What do we mean by ionizable? In the cells, they can give up the protons from their amino groups and the, what we can say, the carboxyl group, and attain a particular charge, okay? Either they may be net positively charged, or either they may be net negatively charged, or they may be having no charge at all, okay? They may be having no charge at all, right? Now, out of all these types, that is, it may be carrying a net positive charge or a net negative charge, the most interesting point that we have to consider is about the molecule that is having no charge at all, right? And the amino acid which is carrying no charge at all is called as the Zwitter ion, right? So let me erase this and let me draw the what we can say, the three different charged molecules or the ways of the amino acid. For example, we are having this one, and alpha carbon and R group, and we are having hydrogen and carboxyl group, this one, yeah. So next we are having one more structure, COO minus, yeah. So we are having R group, and here we are having the H, and finally the last structure that we will be writing is this one, COO minus, H group, and then R group, exactly, right? So this is what they have mentioned in your textbook if you look over here. So now this is an A molecule, consider, and this is a B molecule, and this is the C molecule. And now if we try to look into the net charge that this particular molecule possesses, be it A or be it B, be it C, whatever, okay? So you may expect a question where they will mention about the amino acid. They may draw and give an amino acid, and they may ask you what is the net charge of this particular amino acid, okay? See, the calculation of net charge of the amino acid is way more complex. It's completely, what we can say, out of the scope of the current discussion. But based on the diagrams or the molecules that they have mentioned in your textbook, we can just draw, or we can just come to a conclusion about the charges over here, right? See, now the A molecule is having only one positive charge, but no charge in the carboxyl group. Hence, we can say it is a +1 is the charge of this particular A molecule. But now if you look into the B molecule, the net charge of this particular form of the amino acid is going to be 0, because you see that it is having both the plus charge and one more we are having is the minus charge. Both the charge will get cancelled, hence we say it is the net charge of this B form of amino acid is going to be 0, right? And the last C diagram what we have drawn over here, the structure of the amino acid, there is no positive charge on the amino group, but however the carboxyl group is carrying one negative charge, hence the charge is going to be -1, right? So in this way, from the diagram or the structures that they have mentioned in your textbook, you can expect one question, right? Identify which of the following is a Zwitter ion in this way, or they may even ask you, or they may give the structure of that particular amino acid and they may ask you the net charge, right? So don't worry, they're not going to ask you beyond the scope of whatever they have mentioned in your textbook. Ultimately the questions are going to come from the mentioning part of your textbook itself. Okay, so don't have to bang your head regarding trying to learn advanced concepts. Okay, you will indeed study the advanced concepts as you take on the higher classes. Yes, so this is mainly about the amino acid, right? So out of these three structures, that is A, B and C, the B structure is called as the Zwitter Ion, right? That is the net charge of this particular molecule is 0. I hope I have made quite clear regarding the Zwitter ion, and it is an important point that you people have to remember among the amino acids, right? Examples for the amino acids based upon the charge group or something like that, remember those, okay? And also try to remember about the Zwitter ion. Yeah, so just to try to draw these structures much more and more frequently, you can see that you will try to remember this in a better way. The next one we can look over here are the lipids. Okay, now lipids are actually water insoluble, right? See, we have studied about the lipids, and one of the important physical chemical property of the lipids is that they are completely insoluble in the water, right? Yes. So now some lipids can be simple fatty acids. Okay, some examples for the lipids may be simple fatty acids. Okay, and generally to the R group, you see that the carboxyl group is actually attached. Okay, the carboxyl group, that is COOH group, is attached to the R group, and based upon the number of carbons, okay, based upon the number of carbons including the carboxyl group, an example they have been mentioned over here, that is the palmitic acid. It mainly has around 16 carbons, right? If you have tried the structure of palmitic acid, it is CH3, CH2 and 14 COOH, right? So this is, sorry, yeah, so this is the formula for the chemical formula for the palmitic acid, right? And also we are having one more that is called as the arachidonic acid, and it is mainly made up of 20 carbons. So in the case of both palmitic acid and also the arachidonic acid, remember we are considering both the carboxyl groups over here. Okay, considering both the carboxyl group, it is mainly having 16 and 20 carbons, right? So they may ask you how many number of carbons are present in the arachidonic acid or palmitic acid. Remember, always the number of carbons are going to be even in number, never the odd case, right? Yes, this is one simple way to remember it. Now arachidonic acid, if you try to recall the formula, it is a CH3, similar to that of the palmitic acid, CH2, and we are having 18 and COOH, right? So 18 carbon from the CH2 and 1 from the CH3 and 1 from the COOH will completely make us 20 carbon. Same holds true for even palmitic acid. It makes the 16 in the case of palmitic acid, right? Yes. So now in some cases they may be saturated or they may be unsaturated. For example, palmitic acid is an example for a saturated, and arachidonic acid is an example for the unsaturated we can say fatty acids, right? Now another simple molecule or a simple lipid we are having is the glycerol, right? We are having glycerol as another example that they have mentioned over here. And if you try to look into the structure of the glycerol, it is a CH2 OH, CHOH and CH2 OH, right? Hence it is called as the trihydroxypropane because of 3 hydroxyl molecules present in the chemical structure, right? So it is called as the glycerol. The alternative name we are having is the trihydroxypropane, right? For a propane molecule, we are having 3 hydroxyl groups, hence it is trihydroxypropane in a simple way, right? And glycerol are very much important, okay? So now in the case of glycerol, if you esterify the glycerol, okay, along with the fatty acids, okay, we get some interesting molecules, okay, that we can look over here. Fatty acids are actually esterified with the glycerols, okay, and the result of esterification of fatty acids with the glycerol may yield us mono, not a two, it is a di, okay, mono, di and a triglycerides, okay, mono glycerides, diglycerides or the triglycerides, right? And another type of lipid we are having is the oils, okay, and generally oils are having a lower melting point than the fats, hence in even colder temperatures they remain as oils, right? An example for the oil is the gingerly, okay, sesame oil, we call it as a sesame oil, called as the gingerly, right? Yes. Now we are having one more type of lipid called as the phospholipids, right? Now if you try to recall the term phospholipids, we have mainly encountered this term in the where cell membrane, right? If I'm not wrong, in the cell membrane you have studied about the phospholipids, right? And one example for the phospholipid we are having is the lecithin, or the another name for lecithin we are having is the phosphatidyl choline, okay, phosphatidyl choline, okay, an alternative name for the lecithin, remember this, right? Not required though, but still as an extra point to remember, okay? And why they're called as phospholipids? Because along with the fatty acids, right, plus glycerol, right? So what is the structure we get? Fatty acids plus glycerol, we get actually the glycerides. Okay, remember, if there are 3 fatty acids, we call it as a triglycerides. If it is a single fatty acid, it's called as monoglyceride. If it is made up of 2 fatty acid chain, then it is called as the diglycerides, right? So along with the glycerides, that is the fatty acid plus glycerol, we get the glycerides, right? So if I attach a phosphate molecule, okay, if I attach a phosphate molecule, the ultimate result that you're going to get is the phospholipids. Yes, phospholipids, right? Yes. And one such example for phospholipid we are having is the lecithin, as we just mentioned a moment ago, right? So the phospholipid, that is lecithin, is having one more group called as the choline group, okay? Hence the name is phosphatidyl choline. Okay, so let's not look into the more detailed structure of the phosphatidylcholine or lecithin, right? So now in some cases, that is in the neural tissues, in the nervous system, you do find more complex lipids, okay? The more complex structural lipids are actually found in the, we can say, neural tissues. For example, ceramide, okay? We are having ceramide, or we are having sphingolipids, right? Yes. Ceramide is an example for the sphingolipids, okay? Ganglioside, right? Cerebroganglioside, so on. There are many examples, but generally sphingolipids are mainly found in the neural tissues, and sphingolipid is an example for the complex lipids. Example for complex lipids. See, I hope you people might have heard of the one more term that is called as the glycolipids, okay? Glycolipids, okay? Try to recall which cell organelle is involved in the production of glycolipids and glycoproteins, right? I will not mention it over here, but you people have to try to recall regarding the site of synthesis of the glycolipids, okay? Glycolipid is also found in the nervous system, okay? And it is also an example for complex lipid, right? So some complex lipid structures are also found in the nervous tissues.